From our last lecture video, we know that the overall structure of DNA is a double helix. And we know that our double helix is made up of our 4 nucleotides. So now we're going to take a look at how our 4 nucleotides are arranged together to produce this double helix molecule. So the primary structure of our DNA molecule is a string of deoxyribonucleotides. A nucleotide is the monomer or single unit of our DNA strand. Now, DNA is made up of 4 different monomers that we'll see in just a minute. There is a 5th nucleotide that we find in RNA. We're going to save our discussion of RNA mostly for chapter 10 when we talk about producing RNA, but we are going to talk about its general structure today because it is a nucleotide or made up of nucleotides just like DNA. So the basis of both types of nucleotides, whether it is deoxyribose or deoxyribonucleic acid or DNA, or ribonucleic acid, or RNA. No matter which one it is, it's going to be made up of nucleotide monomers. Now, our nucleotides are made up of smaller pieces as well. The 3 parts of those nucleotides are a sugar, either ribose or deoxyribose. Ribose is a 5-carbon sugar, and deoxyribose is the same sugar, except that it is missing an oxygen on one of its carbons. So if we look at these images here, these are ring structures for sugar molecules. So sugar molecules can be linear, but in solution, they fold back on themselves to make a ring shape. Within that ring is one of the oxygens. So in a sugar, each carbon is attached to an oxygen and a hydrogen. And if we look at each of these carbons here, the first carbon, we've got oxygen, hydrogen all the way around. And then we've got this oxygen, rather than being attached to just one carbon, it's attached to 2 carbons. So when we look at the structure of our ribose sugar, we are going to start by moving clockwise from the oxygen around the sugar ring. And when we do that, the very first carbon that we encounter is called the 1' carbon. And this little apostrophe is, we say that as prime. So 1', 2' would be the next carbon around in the chain, 3', and so on. And our nucleotides are made up of different functional groups that are attached to this ribose base. In RNA, the ribonucleic acid, the 2' carbon has a hydroxyl group. So a hydroxyl group is an OH functional group. And our ribose sugar has that hydroxyl group there on that 2' carbon of our sugar. Deoxyribose has the same structure, except because it is deoxy, it is missing the oxygen on that 2' sugar. So we just have the hydrogen there. We'll see in just a minute that off of the 1' carbon, we are going to have our nitrogenous base, and off of the 5' carbon, which is that carbon that sticks up off of the sugar, we are going to have our phosphate group. Also important is going to be this 3' carbon, where we have another hydroxyl group. And the 5' and 3' carbons are going to help us establish the direction of our molecule. So we're going to come back to those two. So each of our nucleotides is made up from either of either deoxyribose or ribose. We are going to focus on deoxyribose sugar for now because our chapter is going to focus on DNA. Attached to that, we have a phosphate group. Now, the phosphate group is going to be a phosphorus molecule plus 4 oxygens. And we can see that here we've got the phosphate in the center, an oxygen that comes from the oxygen attached to that ribose sugar at the 5' carbon, and then we've got 3 other oxygens in the ring. One of those oxygens forms a double bond. The other 2 should have hydrogen attached, but instead they release that hydrogen, and so we have a negative charge on that phosphate group. The reason that we call this molecule a nucleic acid is that these phosphates release hydrogen ions into solution. A molecule that increases the hydrogen ion solution is called an acid. So molecules that freely release hydrogens are acid, hence the name nucleic acid. Attached to that 1' carbon on the opposite side of the molecule, we have 4 options for nitrogenous bases. Actually 5, but in DNA we have 4 options. We have adenine, guanine, cytosine, and thymine. And then uracil is present in RNA only instead of thymine. So 2 differences now between DNA and RNA. One is the type of sugar and the presence of the oxygen on the 2' carbon. The other is the nucleotides that make it up. So we find thymine in DNA, uracil replaces thymine in RNA. Our other 3 nucleotides remain the same. All of our nitrogenous bases are going to be made up of nitrogen containing rings. And we have 2 different types of nitrogen containing rings. We have purines that are double ring structures. And we have pyrimidines that are single ring structures. And the number of rings is going to be important a little bit later when we start pairing these nitrogenous bases up, and the number of rings across the center is going to affect the width of the molecule. I would like you to know which of the molecules are adenine, which are guanine, which are purines and which are pyrimidines. Adenine and guanine are our 2 purine structures, and then cytosine, uracil, and thymine are the pyrimidine structures with only the single ring. Depicted here are the 4 different nucleotides. You'll recognize that the phosphate and the sugar remain the same through all of them, and the nitrogenous bases are what changes. Even the base structure of the nitrogenous base doesn't really differ between our 2 pyrimidines and our 2 purines. The only difference are going to be some changes in the functional groups that are attached to the molecule, but the base ring structure stays the same. Our purines have a 6 atom ring connected to a 5 atom ring. And then both of our pyrimidines, even though they have different functional groups attached to the molecule, they are still a basic 6 atom ring. And no matter what nitrogenous base, there's going to be some nitrogens contained within the ring structure, all of our different rings in our purines and our pyrimidines. The particular molecule shown here are deoxy monophosphates. We will also see deoxy triphosphates when we get to DNA replication. Our nucleotides actually start out as triphosphates, and then phosphates are cleaved as we link them into long chains of our DNA molecule. But we've got monophosphates that are actually linked in the final structure. So there are two main features to the overall structure of DNA that are going to affect the function of our molecule. Remember, the function is coding. At the most basic level, function is coding. And that is going to depend on complementary base pairing in our strands. And then in order for everything to fit together and for our molecule to be able to code information on both sides, we actually have two strands that run anti-parallel to one another. So anti-parallel is going to be really important when we talk about DNA replication because it's going to affect how the strands are replicated. And then complementary base pairing is going to be important for replication and transcription. It's also going to be important for holding our molecule together. So our overall structure is that we have linked individual monomers. So we've got our phosphate, sugar, and nitrogenous base, and they are linked from sugar to phosphate all the way down. So we've got sugar to phosphate, phosphate to sugar, sugar to phosphate all the way down. They are linked by a bond called a phosphodiester bond. Now, the phosphodiester bond consists of a phosphorus linked to an oxygen linked to a carbon. So that is how we identify that phosphodiester bond. Every single one of our monomers is connected to the next by a covalent bond. In the center, our nucleotide pairs are attached by hydrogen bonds. So hydrogen bonds are an attraction. They are basically a positive and negative attraction between charged regions of two different molecules. So if we look closely at our structure, we have an oxygen off of one ring. On the top of this one, we have a nitrogen off of our other ring. And one is going to generally maintain a partial negative, the other maintain a partial positive. And so that positive negative charge between different regions of those nucleotides are going to attract to one another. Now, hydrogen bonding is an attraction, which means you can pull it apart. Covalent bonds are hard to break. You have to break them on purpose. Hard to break. Hydrogen bonds are basically attractions, positive, negative attraction. And you can disrupt that positive, negative attraction relatively easily. So an individual hydrogen bond is not very strong, but if you get a whole collection of hydrogen bonds all the way along this molecule, it's going to make a very stable molecule. But since they're hydrogen attractions, rather than a covalent bond, they're going to be easy to disrupt when we need to get to our base pairs for replication or transcription. So we've got our two separate strands here. We've got one strand from the top, phosphate to sugar to phosphate to sugar, and so on. So at one end, off of our 5' carbon, we have a free phosphate group. So up here at the top, we've got this phosphate group. We're going to assume for a minute that this molecule this is the absolute end of the molecule. And then down at the other end, we have a free oxygen off of that 3' carbon down there at the bottom. So we say that the molecule runs in a 5' to 3' direction on one strand. Now, if you look at the other strand, we have taken all of our nucleotides and flipped them upside down. Now, this flipping happens because that is how the hydrogen bonds are going to line up appropriately between our nucleotides. And if we look at our cytosine nucleotide here, our guanine nucleotide here, we'll see that there are 3 hydrogen bonds between those 2 molecules. And there are 2 hydrogen bonds between our adenine and our thymine. So regions of the molecule that are GC rich are going to be held together much more tightly than regions that are highs in A's and T's, which means that regions that are high in A and T pairings are going to be easier to open up because we have less hydrogen bonds there. So we've got our nucleotide positioned the opposite direction. It is still from the bottom phosphate, sugar, phosphate, sugar, phosphate, sugar, etc. all the way up. This time we have that free phosphate group down on the bottom and the free oxygen group up there at the top. So we say that they run in anti-parallel directions. So anti in this case, we can think of it as opposite. And parallel means they run next to each other, but they never intersect. So they are parallel to one another, running next to one another, but they run in opposite 5' to 3' directions. The second important part of this structure is the complementary strands. So if we think back to Erwin Chargaff, he noted that the percentage of A in a molecule was always equal to the percentage of T in a molecule, and the percentage of G in a molecule was always equal to the percentage of C. Now this makes sense because when we look at the molecular structure, A's and T's line up very nicely to pair by hydrogen bonding. C's and G's lined up very nicely to pair by hydrogen bonding. And so what we find is that A is always going to pair with T, C is always going to pair with G. The other thing that this means is that we always have a pyrimidine with a purine. So if we look at the top, we've got T, a pyrimidine, with A, that is a purine. We've got G, that is a purine, bound with C, that is a pyrimidine. And this is going to produce 3 rings across so that our molecule always has a constant width. So our 2 main sort of features of our DNA structure are anti-parallel strands running in opposite directions and complementary strands. We'll spend more time on our RNA molecule when we get to chapter 10, but in our RNA molecule, we also have that sugar phosphate strand, sugar phosphate backbone with the phosphodiester bonds in between and the nitrogenous bases coming off the side. The difference is that RNA is going to be a single-stranded molecule, and its secondary structure is going to be based on the function of that molecule. So some of our RNA molecules are going to remain essentially stretched out, single-stranded. Others are going to fold back on themselves based on complementary base pairing and produce a different 3D structure. So for DNA, we're always going to have that double helix. RNA, we're going to have a variety of different structures based on a single-stranded molecule. So now that we have our general structure, our anti-parallel strands with our complementary base pairs, we can zoom out a little bit from our molecule and look at the twists that come up from this double helix. On the right here, we've got our 5' end and our 3' end, anti-parallel, 3' end, 5' end. So we can always identify the ends by what functional group is unattached. So 5' end phosphate, 3' end that hydroxyl group off of the 3' carbon. You'll notice that if we look at this molecule, it's got a fixed width. That is because we always have that purine to pyrimidine binding. You might also notice that it kind of looks like it's a little uneven. Really, it's not. Each one of our twists is going to repeat the same turns at the same angles. And each one of our twists is going to have specific measurements. These twists are based on bond angles that are formed between our nucleotides. And those bond angles force our nucleotides into specific positions. And when they all come together, they form this twist. Now, for one entire twist, there are going to be some regular patterns that we see. For one twist, we are going to have a fixed distance. So the distance between what we call this minor groove here, this little tiny groove, and the next minor groove is going to be 3.4 nanometers. Now this is a very, very tiny, obviously we're at molecular level, but between every full twist, we're going to see 3.4 nanometers. That width that I talked about was 2 nanometers across the molecule from the edge of one backbone to the edge of the next. One entire twist is going to have what we call a major groove and a minor groove. And again, this is just based on our bond angles. DNA forms a right-handed twist. So if you think about screwing in a light bulb or twisting a screw, something like that, when you turn it right, it's kind of what we're talking about with the right-handed twist. Watson and Crick used Rosalind Franklin's photo that turned out to be of a structure called bDNA. DNA has this helical structure, but it has a couple different versions. The two common versions are A and B DNA. The measurements here are the most common type of DNA, which is the B form DNA. The A form DNA is just a little bit different. The overall structure is the same, but the measurements are a little bit different. Basically, the difference between A and B form is that A form is dehydrated. So if you think about if you dehydrate a sponge, if you squeeze the water out of a sponge, it tends to get a little smaller once it's completely dry. When you add water to it, it expands, kind of puffs up a little bit. So our A form DNA is a little bit shorter. Some of our distances are a little bit different. There's a shorter distance between the turns, more nucleotides in one entire turn, and just overall our molecule is squished down a little bit. It's a little shorter and it's a little wider than our B form DNA. There is a third form of DNA, which is a sort of a temporary structure. This is Z form DNA. Z form DNA is produced from a negative supercoil. So we'll talk about supercoiling in the next video, what that actually is. But supercoiling is basically twists that keep twisting. So a negative supercoil is a twist the opposite direction. This form of DNA only happens when RNA polymerase comes through the molecule, and it happens as a mechanism to help open up the DNA. So if you see Z-form DNA in a cell, it's an indication that there is transcription happening. So we can see on the far right, the Z-form DNA, the twists run in the opposite direction compared to that B-form DNA. All right, so just a quick recap. At the beginning of our first lecture video, we said that genetic material has 4 characteristics. It has to contain complex information, have to be able to replicate it, has to produce a physical characteristic, and it has to be able to change. Once Watson and Crick determined the structure of this DNA molecule, they were able to sort of support and explain all of these characteristics that DNA has to contain. So they proposed that the information was contained in the sequence of the nucleotide, so the linear sequence top to bottom. They proposed a mechanism very quickly for replication, which was based on complementary DNA pairings, that as long as you knew one strand, you could make another strand by just using the first one as a template. shortly after their original paper, they published a paper outlining the idea behind gene expression and proposing a very broad general mechanism for gene expression. And this was based on using DNA as a template for an intermediate molecule that could then be used to make proteins. And then because we have sequence of nucleotides that encode information, you could change that information by varying the nucleotide sequence.